Okay, hello. Let's start our demo. Um, we'll first demo the protein structure database, the PDB. And to start with, please open your web browser of your interest, and then you can just search like PDB in any machine. Then you'll get this information of RCSB or the WWPDB. Either works, but I recommend using RCSB because that's what I'm gonna demo today. So click there, and you can see this kind of page. We are gonna learn two things. One, how to search your own protein of your interest, and second, like a general search, like downloading a batch of structure that you want to use. So starting with the first part, we're gonna search some protein of interest. Here I'm gonna show you a demo searching the serotonin receptor. We'll type like a human serotonin receptor 1, for example. But you'll realize this search engine is not very good. So if I type like this, and then you see a bunch of related, but not exactly what you want to find. You can see tons of HT3A, which is not something I'm interested in. This is common weakness of PDB. So instead of doing this, we're going like this. Human serotonin receptor 1 RCSB. Then you'll get its Google search, which is more exact. You click this, you are directed to the RCSB page that contains of your interest, the serotonin receptor of human. So you can do many things, but you can first download things. If you click here, you can check the sequence. You can download MMZIP format, or you can download PDB file. And if you are familiar with using molecular viewers, then you can open that downloaded file like this using PyMol. Then you can take into a deeper look into it like this. But this is out of what you're going to learn today. So we'll just visualize in other ways. And also you can check the experimental data. Because this is EM structure, they provide the underlying experimental data for EM, which is EM map here. You can download that. Or if it's a crystal structure, you can download the diffraction data instead. If it's an NMR, then you can download the NMR data like NOE restraints. So anyways, you can download also the experimental data that was used to model this experimental structure. And we learned about biological assembly, which we called biounits. In this case, because it's a cryo-EM structure, the bioassembly is actually cryo-EM itself. So it's essentially the same. But if it's a crystal structure, by clicking here, you can check multiple different definitions of biounits and also the asymmetric unit that was deposited as PDB file. And if you scroll down, you'll check the classification of this protein, and you'll check the sequence of this protein is actually from Homo sapiens and partially from E. coli. And it also shows where the protein was expressed. It was expressed in some other cell. And it also tells you whether it's mutated from the original sequence. Yes. And then also tells you whether it's a membrane protein or not. Of course, this is yes. And here it tells you who deposited this structure at what date. And the experimental summary is shown here that is determined by EM with the resolution of 3.0, and then how the structure was collected. And if you scroll a little bit more, then you'll see the corresponding paper of this structure you can refer to. This one was published in Nature in 2021 by the same authors. And you can also check related entries of PDB ID that was published together in this paper. And if you scroll a little bit more, then you can find information on each chain or molecular parts. And if you look at here, then it's actually composed of 4 units. This is GPCR, this are one chain, single chain in G protein, other chain in G protein, and another chain in G protein. So individual entry here shows you single chain. For example, this is a GI subunit alpha 1 in G protein. And second is another subunit in G protein. And this is the last part in the G protein. Each are single chains, so they're separate into this way. If you scroll down a little bit more, then you can find the GPCR part here. And this is not the end, but if you scroll more down, because this structure is determined with ligands, you can find the ligand information here. So actual ligand of this GPCR is actually this one, SRO, serotonin. So there's a serotonin bound there. But also you can find other molecules that's determined together. I explained about this in the determination part, that you use other molecules, especially detergents, when you determine membrane proteins. So these are detergent ligand information, which is not biologically meaningful, but which was used to purify the proteins. And that's mostly about the summary part. And by clicking individual tabs, you can get more information. This is a web version 3D molecular viewer. It's not perfect, but you can do quite a good job with this. It tells you hold this 3D structure, and then you can rotate this like this. You can focus, move around. By using wheel, you can focus and focus back. By using right wheel, you can move like this. And by using left click, you can rotate. And if you are move too much, you can click this button. You come back to beginning. And then you can also click like this to see the specific parts in the sequence. For example, if you click, you want to look at the serotonin part, you click this, and then you can see the parts surrounding serotonins. Like this is the GPCR pocket surrounding the serotonin molecule. Okay. And moreover, there are many options here, but you can validate how this structure is good against the experimental data. Like if you click density here, then you can enable electron density map, which shows you how this map, because this happens because we're only looking at serotonin here. You go back. Okay, now we have entire EM structure surrounding the structure. And yeah, this is actually how the experimental look like. There's no protein field originally. And then the experimentalists modeled this protein chain into this experimental density map. So you can see fairly it's a good map. You can see the GPCR part as well as the G protein part here. It's a great match, although you cannot see the atomic details in this map alone. So that's like a limitation of 3-angstrom resolution EM map. Okay, then you can click on the annotation part, and you can see this SCOP or ECOD or PFAM annotations that we learned. But unfortunately, as I explained in my class, that this definition is made per domains. And unfortunately, they don't store all the domain definitions. Like GPCR, this G protein complex contains 4 domains, but it doesn't store all the information. It only shows one of the G protein chain, which is chain B. So that's one of the weakness of PDB website. So we have to explicitly go to SCOP database in order to search for that. Okay. Then if you move on to experiment, you can see the details of cryo-EM experiment on this. For example, you can see how many single particles you were used to construct a 3D image, and the resolution 3.0, and a whole bunch of details that you perhaps don't know. I have to know, yeah. And then here it shows the detailed sequence for each chain. The chain A, which is a first chain in the G protein, and this is chain 2. And let's move on to GPCR, which is the last one. This is the GPCR part, and yeah, it tells you a quite interesting information, like a mutational information from the original sequence. And next thing we are going to learn is advanced search. What if we want to get all the cryo-EM GPCR structures? It's not easy, but we can at least try. So let's try polymer molecular features like this. And then we can say G protein AlphaFoldDB receptors. If you want to specifically look into those that are determined by cryo-EM, click here, methods, experimental methods. You can click electron microscopy. And you can also provide more details, like reconstruction resolution. If we want to look at, oh, okay, let's try before applying this before. By clicking this, you can get the 321 structures. Let's try crystallographic structure instead. Then you have 145. So you have about 500 GPCR structures deposited in PDB. But if you apply this filter, like if you want to remove all the bad resolution ones, oh, this doesn't work because it's cryo-EM. Let's go back to cryo-EM and then apply. And then of about 320, most of them are passing these filters. You can also reduce this into even higher resolution like this. Then there are only very few those match this high resolution cutoff. So you can do a similar thing on like kinases. And you can select solution NMR. This doesn't work. Then you can find 400 NMR structures for kinases. Yep, and also you can click onto it. And then, yeah, same story. You can look at how this works. Of course, you can search for crystal structures for this. And yeah, there are many crystal structures for kinases. And the last thing I'm gonna teach is this how to learn to use PDB. I explained a little bit, but it's much better practice to use this PDB 101. It contains many materials that I taught in the lecture. Like you can click something like this, and then it contains all the information and even more than that about how this PDB is organized and how you can interpret the entries from PDB. Like we talked about missing coordinates, we talked about biological assemblies, and how the structures are modeled, and so on. So it has plenty of data and material that you can learn from. Okay, then let's learn how to use AlphaFoldDB. It's same thing. Google search works the best. You can just search AlphaFold database, and the first part is AlphaFoldDB. And yeah, this is where the search goes. You can type into many different ways, but again, you have to be careful because sometimes it's not something you want to search. Let's say like a human serotonin receptor 1. Okay, so if you type like this, you see it's not exactly matching what you want. So again, you can do it like this. And then the best thing is to search against UniProt. Yep, or this one, yeah. And then by doing this, you can get the exact ID, which is called UniProt ID. Copy this here, and then you'll get exact match that you want to looking for, like this. And then this is the model structure that AlphaFold made for this human protein. If you carefully look at here, the important part is the confidence of this structure globally and locally. So you can see that some parts are like red here, some parts are blue. So blue part means it's confident part. Red part means it's non-confidence. So or usually this red line, red part or yellow part means it could be quite flexible or disordered. So besides this disordered part, the most of the parts are quite good. So you can say this is a quite confident model structure. And another way of looking at that is this PAE value. So it's green at the region where it thinks is confident. By the way, this is a pairwise metric. So it tells you the distance between this y-axis and x-axis is confident when it's closer to green. And then you can see this part from residue number 30 to about 240. It's quite confident by itself. And then from here to here is very non-confident. And again, this part is confident. And then if you look at in this structure, this part corresponds to first missing part, and this part belongs to this part. So you can say by trimming and this flexible region, you can trust this structure is accurate. And of course, you can download their files either in PDB file or mmzip format, or even you can download the confident metric provided by AlphaFold too. And you can do more than this. If you click E. coli, then you can see all the proteins that AlphaFold modeled for E. coli. And it contains this many proteins. And then you can go to download part. By clicking one of these, you can download a group of structures that AlphaFold predicted. For example, if you're interested in all the protein structures of human, then you can download this. And same for other organisms. And there are many interesting other parts like this. You can download all the prediction files. And if you're interested in more information, then you can click here about details of how this method works, how this missense prediction was made by alpha missense, and many information that you might be interested in.